All right, we're back here, plenary session, European edition. I'm joined by Dr. Timothy Olivier. Timothy is, of course, a oncologist based in Geneva, Switzerland. Timothy, it's a pleasure to see you.
It's a pleasure for me. It's a pleasure to have you. So it's you are my guest today. It's a Swiss Alps edition.
This is the best type of plenary session. This is the Swiss Alps edition, and I gotta say,
Absolutely,
it's a good edition. It's a good edition, much better than Zoom edition. Much better
much
than
better,
Zoom.
much better.
Much better. And we've been busy. Recently, I came to your hospital, University Hospital Geneva, and gave a lecture to the oncologists, and I thought it was, just a really great discussion.
Yeah, it was very engaging. I really like it. The talk was great. Actually, it's a lot of new stuff you presented there, and the following discussion was very engaging. And I think the feedback were very positive.
That's good to hear. Yes, I made new slides. I make new slides when I'm on airplanes. That's how I. That's when I make my new slides. So we have a lot to talk about. We have a number of new papers that Dr. Olivier has published, and we're going to run through those. But I see that we're in Europe, and the World Lung Cancer is going on right now. World Lung Cancer in Vienna, Austria.
Yeah, not far from here.
Not far.
Yeah,
But they look hot. They don't look like they have the cool European, the Swiss Alps kind of
maybe
feel.
the altitude here is cooler.
Yeah, the altitude here is a little bit better. But they have a lot of altitude with the bombastic rhetoric that I hear coming out of the conference. So I saw something very interesting. The IMPOWER010 study. This is the randomized control trial of atezolizumab in the adjuvant setting. And of course, we had disease-free survival, but now they have published the overall survival. And they have stratified it based on based on TC PD-L1 stain. And what they show is that if your PD-L1 50% or higher, there appears to be a clear overall survival benefit. And if there's above 1%, which includes the 50% and higher, there's a trend to it. They call it a trend towards a benefit. There's a trend towards a benefit. But of course, if you just looked at 1 to 49, it looks like there's no benefit at all. So you've been following this. What are your thoughts on
Yeah,
this?
I've been following this. Maybe you follow more closely the very recent presentation in this lung cancer summit, but there's a lot of change in the neoadjuvant and adjuvant space in lung cancer. I think this is interesting, and we just published a commentary about the CheckMate 816 trial in the neoadjuvant space that has several limitations. Also, I can imagine that many people are very excited by the results. We highlight major concerns. Maybe we can go through that.
Yeah, let's go through that.
And this was, I think, IMPOWER010 led to the FDA approval of Atezo in 2021 or
Right. A
already.
couple years ago,
Yeah,
yeah.
yeah, yeah. So I don't know what are your thoughts. I know you published something about the nested and adjacent subgroup. I think this is one issue maybe you want to talk about.
Yeah, I think that that's one of my big issues, which is that, it's not helpful to me to tell me how the outcomes do in above 50% and then above 1%, because above 1% includes above 50%. So I want to know 1 to 10%, 11 to 20%, 21 to 30%, et cetera, et cetera. In this case, 1 to 49% looks totally null, but it would be nice to see some breakdown even within 1 to 49. It's a big bucket, and then above 49%, it would be nice to see breakdown from 49 to 59 and 60 to 69, to break this out into more sort of concrete subgroups. And so we call this nested subgroups, where one is within the other, and adjacent subgroups where they're next to each other. And you will learn a lot more with adjacent subgroups. Now, of course, this PD-L1 stain, it's not a perfect stain. And if you stained it and I stained it, you might say 62% and I might say 42%. That's fine. We can have that discussion about the inter-rater reliability. But at a minimum, I think it's important to know, however you slice it, is somebody who is, 10, 20%,
yeah.
90%, yeah, or 90% benefiting.
Yeah.
Is a 90% benefiting the same way as a 40%? And, sometimes it is kind of obvious. And so that's my concern here. The other concern always with these kinds of studies is something that we've been working on, which is if you are on the control group, some of those people will eventually relapse, and they should all get a checkpoint inhibitor. They should all get a checkpoint inhibitor, probably in the frontline setting of metastatic disease. How many of them did get a checkpoint inhibitor? And that will have implications for OS.
I think, yeah, that's a good point. We have a work on that. I hope it will be out soon. In the CheckMate 816 trial, we just exemplified it. Just remember, all patients are eligible for immunotherapy. So there's no reason that these patients in the control arm, they won't get immunotherapy if they relapse.
Yeah.
And in the CheckMate, so just a quick reminder, CheckMate 816, it's neoadjuvant lung cancer, early lung cancer from stage 1B, I think, to 3A.
They have to be thought a surgical candidate at the outset.
Yeah. and 3 cycles of either chemo or chemo plus nivo. Then surgery, and then they could have adjuvant chemotherapy and then surveillance.
I see.
And for those patients who relapse in the control arm and receive a systemic treatment, 65 of them receive immunotherapy. So 35 did not receive immunotherapy. I think it should be 100% because they are all eligible for, because they are in the trial and they are fit.
Yeah.
So I think
That's
as
a
you
huge
said,
limitation.
Yeah, huge limitation.
I want to run through your 4 limitations, but I think that's a huge one. But the other thing I want to point out is that before this study existed, I didn't say neoadjuvant often in lung cancer. It wasn't a thing we talked about.
Yeah.
And I've asked people, what is the best study that you're going to hang your hat on to show me that neoadjuvant is as good as adjuvant? And what they say is generally, well, we know that to be true in other situations like breast cancer. I'm like, well, it's a different cancer. I don't know what to tell you. It's a different cancer, so you need to generate data for this particular cancer. But let's hit your 4 concerns. What were the 4 concerns with CheckMate 816? This is a translational oncology paper.
Yeah, so we can go no matter the order, but first maybe this point, the control arm. So the neoadjuvant chemotherapy is not fully inappropriate. We don't say that, but the mainstay, the most common treatment is adjuvant chemo in those
Particularly
patients.
like a stage 2 patient.
Yeah, and you have this LACE meta-analysis, more than 4,000 patients, so it's strong. You have a 5.4% benefit after 5 years of follow-up. And for the neoadjuvant trials, there are fewer trials, fewer patients. Some of them were stopped during accrual, so it was not the standard of the most common standard of care. So there is an issue there, as we said in the paper. We have an issue that because you have this benefit of nivolumab, we will implement systematically this neoadjuvant strategy, but maybe it's not the most appropriate strategy.
I think the answer is we just don't know. And people keep saying that, well, in breast we have many studies that have suggested that there is no difference between adjuvant and neoadjuvant. And all I have to say is in breast you also have hormone therapy. We don't have that in lung. It's a different cancer. It's a different cancer. It's a very different cancer. And so you just don't know until you demonstrate to me. And what will that take? I think it would be a large sort of meta-analytic kind of approach, but I just don't think we don't even have the studies to put in the meta-analysis to ask if they're comparable.
I think the comparison with breast is a good transition to another point we
Okay,
made
point 2.
about the primary endpoints of the study.
Yes, okay.
PCR, pathological complete response, and EFS, event-free survival. And again, a lot of people are talking about breast cancer, but PCR under immunotherapy in the neoadjuvant setting is not, even an EFS are not validated surrogate.
In lung cancer.
In lung cancer, I think it's very important to understand that a surrogate is validated in a very specific condition. It can be validated in breast cancer under chemotherapy and no longer be valid in other conditions.
And I want to add that it is not even validated in breast cancer. What are we talking about? We just lived through the experience. One, we used PAT-CR for the approval of adjuvant and neoadjuvant pertuzumab. And that was done based on a meta-analysis by Pat Cortazar and colleagues in The Lancet, which showed that PAT-CR did not predict even EFS, and EFS doesn't predict OS, and PAT-CR doesn't predict OS, so these things just don't predict each other. And of course, we have to back up. One, what is the goal of adjuvant therapy? The goal is, you cut out the cancer, you give some drugs, either neoadjuvant or adjuvant, and the goal is that that person is cured of the disease. And the goal of adding more drugs is to increase the curative fraction. The goal isn't to delay the time until there's a radiographic appearance in the femur. The goal is to increase the fraction of people in whom the cancer never comes back in the femur. That's the goal, is to cure more people. And so the question is PAT-CR. What's PAT-CR? You cut the tumor out, you look under the microscope, you can no longer see disease in the breast or in the nodes. That's scored as a PAT-CR. There's no doubt about it, it's prognostic. It's better to achieve PAT-CR than not achieve PAT-CR. But a prognostic marker is not the question of surrogacy. Surrogacy asks if, in the aggregate, in hundreds and thousands of people, I increase the fraction of people who achieve a higher PAT-CR, do I also increase the fraction of people cured years down the road? And that's a different question than for the individual who achieves PAT-CR, do they do well? And the answer is, yeah, of course they do better than the person who doesn't. That's a separate question, that's a prognostic question. But we were asking the surrogate question, can we hang our hat on this? And I think the answer in breast is it didn't do that. And we finally had the results of the long-term OS follow-up of Affinity, which showed that pertuzumab isn't improving OS. What are we talking about? So PATCR failed in breast cancer. It was a huge regulatory blunder. I mean, it led to the expensive use of a costly product that has toxicity, didn't increase curative fraction in breast, and now you're bringing over to lung where you don't even have that. You don't even have that data.
Yeah.
What are we doing? What are we doing?
And yes, and I think the point we are making is it could have been approved on the accelerated pathway, but it was approved on the regular pathway. And this is also a point because normally the accelerated pathway can provide a safeguard until you have the overall survival result. But here nivolumab was approved as a regular approval. Yeah.
CheckMate 816.
Yeah.
And one point to say here, I think, is that many people will say that DFS in the adjuvant setting has a correlation in lung cancer with OS, and I think that's true in the paper that Allison and I did in the European Journal of Cancer in 2017.
Under chemotherapy.
That's it.
Under chemotherapy.
That's it. He knows where I'm going. Under chemotherapy. In other words, that relationship, that 3-year DFS correlates with 5-year OS in lung cancer in the adjuvant space. One, it's not the neoadjuvant we're talking about here, it's adjuvant. 2, it's for chemotherapy drugs. It's not for checkpoint inhibitors. Now, why might checkpoint inhibitors be different?
And before we go to the other points, my worry is that my worry is not that the overall survival result will be positive. I think maybe they will be positive. I don't know.
Sure.
But if you don't provide adequate post-progression treatment,
yes.
If those patients do not receive appropriate immunotherapy when they progress, they could be a surrogate validation, but it's not really what you will have in your practice.
Correct.
So I have a worried here, and I think the main difference is that in the metastatic setting, you can derive long-term benefit with immune checkpoint inhibitor, even when you are in the advanced or metastatic setting, and that was not really the case with chemotherapy.
No, I think that's the key. That's a key point, which is that in order for these surrogate correlations to hold true, there has to be certain principles of the drug. And cytotoxic drugs in the adjuvant setting can increase curative fraction by eradicating microscopic disease. That same drug in the metastatic setting in lung cancer, a cytotoxic drug, can never cure anybody. So it works here, doesn't work here. With immunotherapy, we don't know that the effect in the adjuvant setting is fundamentally different than the effect in the metastatic setting. It might work really good in the metastatic setting, which, if anything, means that the DFS is less predictive of the OS later. And then your point is excellent. One of the 4 points, I think we've hit 2 of the points. Yeah. 1, we don't know about neoadjuvant. 2, we don't know about PATCR EFS. But the another point is that when you finally come with positive OS, and I'm sure they will, because 35% of the control arm is getting delinquent therapy. Yeah. So they will have a positive OS, because it's better to get a checkpoint inhibitor now than never. Yeah. But I didn't need this study to know that. I know that to be true from Keynote 189, etc. But that doesn't prove or vindicate this strategy.
Yeah. I mean, that doesn't prove. It's more about the external validity. When you will be practicing with optimal control arm and with optimal post-progression treatment, when you have full access to all these drugs, it's not sure you will have the same outcome.
And I think people we call that external validity, but really that's like, does it apply to your practice?
Yes, to your practice.
Your practice.
Just be simple. Does the trial apply to practice? And I think it's a basic question in critical appraisal that answer many questions.
Yeah. Okay,
I think we made 3 points because there was the primary endpoint, there was the post-progression treatment, there was a control arm. Yeah. And the last point that was a bit tricky is that in this trial, there were 8 amendments of versions. So they changed the protocol or version or statistical plan 8 times. And it's very difficult to appraise why they did all that thing, because they are redacted.
It's like the Mueller report. Yeah.
And our concern here is that they could have changed the statistical analytical plan by knowing some of the results, or maybe for strategic purposes. We don't know.
Recently I had it reminds me of something. I was writing a document, and in the document I talk about a trial, and I'm not going to get into the details because it's a separate issue. But I said that the company and the FDA allowed them to make a major protocol amendment where they're basically going to do something totally different. They did what they said they would do. They didn't achieve the endpoint they thought they would. Then they were allowed to totally modify the study and try one more time to get the endpoint they wanted. And I think it's so problematic. But somebody who read my draft, they said, explain to me why this is problematic. And I said, well, it's very difficult to explain, but let me do my best. Let me try to explain it. And I was thinking about it, and I was like, basically you did what you said you would do when you had failed, and then you get one more try. But the thing is, now that I've granted you one more try, do I really know that one more try is all you would get? I don't know that. In fact, if anything, if that try didn't work, you get to try again and again and again and again. How many times will you be allowed to try? And then you get into the problem of the more you try, by chance alone, eventually there'll be something that looks kind of close. And this particular trial had a non-inferiority design, so they're just trying to squeak in above the delta or the margin. And so I know that if you get try after try after try after try, and your non-inferiority margin is big enough to park a school bus in, you're going to get a victory eventually. And so it really, trial modifications can be done right. They can be done in a good way where people think about the alpha error and they think about all these things and they do it in an honest and clean way without looking at the data. But trial modifications also can be misused and abused. And the only way to tell those two apart is if you knew why they did what they did when they did it. And you don't, because this thing's redacted.
For just to give an example, the primary endpoints were modified to become
PATH-CR and EFS.
PATH-CR and EFS.
What was the original? OS.
EFS and OS. I'm even not sure.
Okay.
You have an initial arm of Nivo-IP that was closed.
I remember that.
You have a lot of things, and maybe there are some things that are fully justified, but the issue is that we can't really appraise it.
The only metaphor I think of is the Texas sharpshooter fallacy, which is that if we go to a barn and I give you a pistol and I say, shoot at that target that I've painted on the wall, I can judge whether or not you're a good shot. But if I let you shoot at the wall and then you get to go paint the bullseye afterwards, you're going to look pretty good no matter how bad you are. And that to some degree is what happens when you allow all these kinds of protocol modifications. You're committing the Texas sharpshooter fallacy. You're letting the person just shoot and then say, you know what, actually, let's just look at that PATH-CR. And after all, they're actually saying, because we use it in breast cancer. But they forget to say that it actually doesn't work in breast cancer. You shouldn't have used it there, so why the hell are you using it here? Okay, that's a great point. So these are the 4 points. The paper is out where? Translational Oncology?
The paper is out in Translational Oncology with a graphical abstract that is just summarizing the points. And again, it's just not, it's not being critic just to be critic. It's that it's not applying to our practice. I think that's the main point that we are making about the control arm, about post-progression. And maybe it's a really transformative drug in the neoadjuvant space, but I have some doubts. We remain with doubts.
Now, I think that's really well put, and people should read the paper. I interviewed Jack West on a prior episode of this podcast, and we talked about this, and he said the only thing that I will say is in its favor, which is it was a fixed course of nivolumab. Isn't it nice that you're 3 and you're done? You're 3 and you're done, rather than and in
for
fact,
instance, 1 year of adjuvant.
But
it's a bit frustrating to have many trials when you see many limitations and you have to compare these and make your choice, when you think you could do those trials with proper control arm, proper post-progression treatment, proper statistical analysis, pre-registration, all this kind of thing. And you don't have them.
That's well put. And I just want to put the problems in 2 buckets. The problems we are talking about that you have enumerated as these 4 problems, these are problems with this study that our current regulatory law allows the FDA to correct. They are totally free, if they so choose, to ensure, one, we're going to be very reluctant to give you a neoadjuvant approval until you have validated the neoadjuvant strategy. They are very free to say, we will make you have OS as the primary endpoint because our goal is curing people, and you gotta give checkpoint inhibitor post-protocol. That's within their current legislative authority and their regulatory law. They can do that. The one thing they can't do under current law, which I would like them to someday be able to do, is to say, we know BMS is going to run this nivolumab CheckMate 816, and we know Genentech Roche is going to run IMPOWER010, and we know that all these companies are running these kind of similar trials. We should harmonize it, create sort of a single multi-arm recovery platform.
That would be great.
That would be great. But that would take a law. Congress has to change.
The last thing, for instance, in IMPOWER, IMPOWER010, I think they excluded EGFR and ALK, and not in CheckMate 816. So you see, there are some discrepancies in the inclusion exclusion criteria. So it's very difficult to just interpret all.
I swear I just saw a slide.
Maybe it's reverse.
Maybe you flipped the two.
Yeah.
Yes.
We have to double check.
Yeah, because I swear I saw a slide where they showed IMPOWER10 with
Okay. I flipped the two, but yeah.
But this is also an important point because now they're presenting
and we
their
have
data.
Adora.
Yeah.
So what we will do? Okay.
No.
Go on.
Well, that's a good point. But I was just thinking that they have the intention to treat population. They have the over 1% population. They have the over 50% population. They have the 1 to 49% population. They could present the results with the driver mutations or without the driver mutation. They have so many degrees of freedom that of course they're going to get some wins out of this.
Yeah.
You know, and the Adora point I think is the same points. I mean, the problem with Adora is all the DFS-OS correlation does not apply to TKI. The second problem is we don't know post-progression if the control arm is going to get osimertinib per Flowra or not. They ought to. It's the same sorts of things. I don't know how do you weigh these things. I mean, the differences are this is a lot more people. So the amount of money per annum is going to be a ton.
Yeah.
Adora is a lot more years, though, isn't it? 3 years of therapy, and it's not a trivial. I think I made the calculations in many weeks ago. Yeah, millions of dollars to even avert an event. You know, I think they could do better studies. Simple as that.
Yeah.
What's the next paper that you want to go to the next paper?
The next paper, head-to-head, so drug dosing and GCSF paper.
Yeah, let's do
European
it. Okay.
Journal of Cancer. Do you want to talk about that?
Yeah, and I guess I would say that it's one of those papers that, you know, I don't know how many people are out there reading papers, to be honest. Everyone wants to listen to a podcast. You know, that's all. Nobody's reading papers, but there are a few people who still read papers, and one of those people is Mani Mohyuddin, the great myeloma doctor, and you know, I saw him tweet about that paper, and he said something like, Timothy, you know, I don't know if you saw this tweet. Something
I'm not sure.
He said, Timothy, this is like, you know, a super excellent and thought-provoking paper. And yeah.
From Mani,
that's high praise, huh? Yeah. It's high praise from Mani. And I totally think that's true. And it's one of the few papers that you just genuinely get excited about because after a while, you publish a paper, then, you know, lots of people start to say something similar, which is good because that's what it means to change the field. That a lot of people are saying something, but you know, also it's also like, you know, I don't know, lost the novelty of it. And, you know, I feel like it's been said and done. And so I want to move to a different idea. And when you publish this paper, it's just something that not a lot of people ever talk about.
Yeah.
I never heard anybody talking about it.
Yeah.
So just tell us about it.
So actually, you are maybe modest because you published about the same topic in 2014.
Yeah, a little, but not the same way.
Yeah. So, but this issue was raised. So, maybe some background.
Yeah.
So, what we studied was the rules in the protocol of two things: the drug dose modification. So, when you have toxicities or when you have some rules, and sometimes you can lower the treatment dose, sometimes you can higher the treatment dose. And we wanted to compare those rules between arms. And the other thing we studied was the GCSF rules. So GCSF are colony stimulating factors, and it's mainly for chemotherapy when you have the neutropenia that can induce severe infection. And so it's used in patients undergoing chemotherapy, either prophylactically or curative intent, just to higher the count of neutrophils and to reduce the severity of infection, we can say.
That's perfect, yeah. And then to be fair, that's something that I've never published on.
Yeah, so the GCSF part is really a thing I thought about at the initiation of the project. So the project was your idea, and it came from the work you did with Tito Fojo. So this was described since 2014 at least,
That's the only time,
but in some trials there were some unfair rules between arms, favoring the experimental arm often, but it was not really counted how often it happened, and when it's the experimental drug, when it's the control arm. And so that was the purpose of the paper, just to systematically appraise head-to-head trials. So we went from 29 to 2021, FDA registration head-to-head trials where you could compare those rules.
Yeah.
So we ended in 62 trials.
Yeah. But before we get that, let me give one example.
Yeah.
That's,
Yeah, give an example. Yeah.
one example so they can kind of see it. You know, and I think this was the example that got Tito and I to think about it. And this was when I was at the NCI, and Tito, of course, was program director there. And the example was AXIS, axitinib versus sorafenib in second-line kidney cancer. It was a provocative study because I think, to my knowledge, it was the first trial in second-line kidney cancer that ever took two active anti-cancer drugs, pitted them head-to-head, and found a PFS benefit for one over the other. That was axitinib. Had a better PFS than sorafenib, and for a while it kind of changed standard of care. Of course, standard of care has been rewritten many times, and now, of course, you know where we are. We're with checkpoint inhibitor plus Axipam, and Nivo, Cavo, and everything you can think of, right? But back then, this was exciting stuff. It wasn't even that long ago, you know. We published our thing about it in 2014. I think this came out in the year 2009, 2010-ish. Brian Rainey's paper in the Lancet. Axitinib versus sorafenib. They're both two dirty TKIs. They're both TKIs that hit a lot of the tyrosine kinome. They're not very specific. They're dirty drugs, but they both sort of slow the growth rate kinetics of kidney cancer. We looked at this study. There's a PFS benefit, and we noticed one thing about the dosing. The dosing of the drugs, of course, sorafenib was stuck with that original dosing from HCC, which is 400 BID, then 400 daily, then 400 every other day. So in other words, if you took it 400 BID and you had any toxicity, you were told to dose reduce to 400 daily and then 400 every other day, which is a 50% dose reduction, and then you're taking a quarter of your starting dose in 2 dose reductions. The other thing we knew about this study and sorafenib was that the rates of dose reduction, they're not 1%, 2%, 4%, like some of the old studies of cytotoxic drugs. The dose reduction rates are 37%. 47%, 22%. You know, there's significant fractions of people getting dose reduced. And so the dose reduction was occurring frequently, and the step size was large on sorafenib. Then we look at the step size on axitinib, and it's not 50%, 25%. If I recall correctly, it's 66%, 33%. And if you were doing well on axitinib, you could dose escalate twice. Twice. You can crank up the dose. And 30-some%, about a third of them, were actually dose escalated. And then there's a third thing, that the rule for hypertension, if you had the hypertension rule was different. That, in other words, if you took axitinib, and one of the class effects of these drugs is it causes high blood pressure, you were allowed to give more antihypertensives before you were required to dose down, and you could crank them up with some antihypertensives on board. But sorafenib doesn't get that. Ultimately, it has this PFS benefit. But the real question I have is axitinib a better molecule than sorafenib as a molecule? Or is it the fact that many people get dose reductions, these dose reductions are shallower, these people get dose escalation, and we do know that drug dosing is sort of this idiosyncratic thing where there's some people that they only benefit from cranking the dose, other people that they're deriving benefit from the minimally effective dose, and having more sort of steps, having smaller steps gives you a little bit more flexibility in trying to pushing the dose up. And okay, so we noticed this. We wrote a paper in, I think, called Oral Anti-Cancer Drug Dosing in the JCO, a Commons and Controversy, but we had never sort of systematically looked at all these studies.
How often it occurs.
Okay, so back to your paper.
And so it was very interesting because finally, at the end of the day, it occurs to have unfair rules in 65% of trials. So 65. So, okay, now it could be fair if it's sometime in the experimental group, sometime in the control group, but 55 are favoring the experimental arm. So 55% of head-to-head trials are favoring the experimental arm in rules of drug dosing modification or GCSF rules. And 10% are favoring the control arm.
I see. So I guess, to be fair, one fair point is that many trials for drugs don't even have a control arm. So your study doesn't apply to that.
So we had to restrict where we could compare.
Yes. Yeah. So the trials that don't have a control arm, they're not in your study. And the trials where I add CheckMate 816 wouldn't be in your study either, because it's the addition of a...
because we excluded, for instance, if you have just a checkpoint inhibitor, you don't have rules of drug dose modification, and you don't have GCSF for checkpoint inhibitors, so we don't use it.
But also, you don't include trials where it's an add-on drug. In one arm, they get an add-on, and the other arm, they don't get an add-on. That's not included. You're only looking at trials where they're head-to-head, and they're different drugs being given, where the dose modification makes sense.
But for instance, if in one arm the GCSF administration may be important, For instance, in our manuscript, we give the example of Keynote 42,
Yes.
Pembro versus chemo.
Yes.
And in the chemo arm, you could receive carbo AUC5 plus pemetrexed or paclitaxel 200 milligram per square meter. And GCSF, prophylactic GCSF was not allowed. So we included that. And
Yes.
we gave the,
But
was
that,
not allowed.
but I guess that I think that it makes sense to me
Yeah,
why one
just
would
to give an
include that.
example. Just to give an example.
Because it seems like it's punishing the
It's
chemotherapy. It's punishing
yeah,
them.
you will maybe stop the treatment earlier because you are not allowed to give it prophylactically for the next cycle, for instance.
Maybe let's separate these two concepts for a
yeah.
moment. The drug dosing concept and the GCSF concept, there's a similarity, and the similarity is that you're not going to read this in the abstract, and even in a journal club, no one is going to say this. This is something hidden in the paper. And what's the hidden thing? It's that this is a bias that penalizes the control arm, doesn't penalize the experimental arm as much, or in fact rewards the experimental arm, which means that you're not looking at a clean test of whether or not the new drug is better than the old drug. It's also the new drug plus a slightly different rule or a slightly more favorable rule versus the old drug. And so, does it matter? Some of these new drugs, they probably are better than the old drug.
Yeah.
But some are probably not.
Yeah.
It's just the dosing.
I think a good example is Lenvatinib versus sorafenib in the non-inferiority trial. I think you describe it in the first paper.
Yeah, talk about
But what is interesting, so the first and second step of dose modification are not the same, and they are favoring the Lenvatinib arm, so the experimental arm. There is also a third reduction dose allowed it in Lenvatinib, but not in sorafenib. So it's a second advantage of Lenvatinib. And what was interesting, we have the result of the dose. How you said?
Dose reduction and the cumulative dose given.
Cumulative dose. So dose reduction was the same.
Yeah.
But cumulative dose is
higher.
88% in Lenvatinib and 83% in sorafenib. So it's a kind of proof or example where you have the same dose reduction but not the same cumulative dose.
Yes, it's a great example. So this is Lenvatinib versus sorafenib, non-inferiority study.
Yeah.
The upper bound was 1.08 hazard ratio, which corresponds to like a 40% loss of efficacy of sorafenib. By the way, if you're taking sorafenib for HCC, I don't want to tolerate any loss of efficacy. It's like, not that great a drug anyway.
The paper of Stacy.
So we don't want to tolerate any loss of efficacy anyway, but this is tolerating loss of efficacy. And Lenva has 3 dose steps, sorafenib just has 2. Dose reduction is common, and even though the dose reduction was occurring sort of at the same rate, or if anything, Lenva had more dose reduction.
It was
comparable.
In this paper, it was 37 and 38.
Okay,
so
it's like practically the identical number.
So that was interesting because it was the same.
Yes.
But the cumulative dose not the same.
Yes, and then there's a metric called like the percent of the cumulative dose you intended to give that you actually gave, and in Lenva was 88%, and the other arm was 83%, which means they're getting less of the dose than you intended to give. So that's a great example of. So I mean, what does it mean? It just means that like, do I really know? Well, first of all, is Lenva as good a molecule as sorafenib? The answer is, well, it's a non-inferiority study anyway. It wasn't the superiority study. But if anything, Lenva might be a worse molecule and just better dosing around sorafenib.
That's possible, yeah.
Now, I want to say, I presented this paper at a UCSF conference, and one of the things people said to me was, and this is a fair point, and I think we say this in the manuscript, which is that, when it's not my job to like fix the drug label on sorafenib, that's the way the label is. I just follow the label. And I want to say that's probably true. It's not it's not your fault. Yeah. But I think companies are smart, and the reason you building in your small steps and you're changing your pill is because that that's a way you can exploit it.
We thought about that, but at the end of the day, 55% are favoring the experimental arm and 10% the control arm. So that is telling. It shouldn't be like that.
That's another good point, right? Which is that if this was just occurring at random, it would be a split. Yeah. Now, talk about the GCSF for a second.
So
is it the sacituzumab?
GCSF, we exemplify this with sacituzumab govitecan in the ASCEND trial in triple-negative breast cancer. And as you said, in this example, you have to really dig in the protocol. Even the protocol and the appendix is not the same, and it's not the same as the FDA labels. So you have to take time. And as you said, in Journal Club, it's not always like that. So we show in this trial that, for instance, first episode of grade 4 neutropenia, sacituzumab govitecan, you receive mandatory GCSF. In the control arm, it was at the physician discretion. So you have different rules like that. And so we studied that, and we found the same kind of findings as dose reduction. So it was favoring the experimental arm. We gave the example of also Keynote 42, like that, where, for instance, prophylactic GCSF.
But let me ask you this.
Yeah.
Does GCSF allow you to push the dose of a chemo drug?
Yeah.
Yeah, of course. Yeah,
Yeah.
that's what dose-dense ACT is, right? No, maybe
explain more.
No, I think like that's an important thing that maybe we take for granted because we know
right,
it,
you're
right?
right.
But giving people GCSF allows you to crank up the chemo,
Yeah.
and we know that to be true because we have dose-dense ACT, and everyone loves dose-dense ACT, dose-dense ACT, dose-dense ACT. You know, do they like TC? They don't like TC as much. I don't know why, but they don't. But they like dose dense. But dose dense, if you took away the GCSF, you'd never get through it.
Yeah.
And many drugs, if you took away the GCSF, you never get through it. And what Timothee is saying is that, I'm fine to let you give it or not give it, but it should be the same in both arms.
Yeah.
If one arm's allowed to get it prophylactically, like everyone gets it before they get the chemo, and the other arm is not getting it prophylactically, well, then you're advantaging the other arm. And if one arm is allowed to get it, but there's some trials where they're actually prohibited entirely.
Yeah, prohibited entirely sometimes, yeah.
But that's ridiculous. I mean, that's like telling me how to practice. I mean, that's like interfering with the practice of medicine.
And it's really, in some little space of the protocol, you find that
it's not publicized. Yeah. So I think, there's a reason why it's not publicized. People weren't looking for it because, to be honest, before these papers on the topic, no one ever talked about these issues. This is the first paper I think that puts GCSF on the map, with the exception of our commentary. And the drug dosing, after that 2014 paper in the JCO, people didn't talk about it too much. They just probably realized it was a great place to exploit. So it's very interesting. And the paper's out in the European Journal of Cancer.
Yeah, European Journal of Cancer. So you, Alison Aslam, and me. And it's in open access. So
that's good.
Yeah, that's good.
So they can actually read it.
Yeah.
Not like...
So no excuse.
No excuse. What was it that I always see people complain,
I know.
You know my paper? Yeah.
Which John,
Yeah. I published the paper and they say, I can't even read this paper. But ironically, the paper, which was about, sort of constructive versus obsessive criticism in science, met with a response that proved the point of the paper, which is that people aren't reading and engaging the material. Anyway, but it is open access now. That paper is also open access.
Yeah, I saw the tweet.
So, what have we covered so far? These are two things. One, CheckMate 816, IMPOWER010. This is the real world lung cancer right here. This isn't Vienna, but it's not too far.
Not far.
Not far.
I mean, by plane or by a bird could go very fast.
We could fly right up, but we're just far enough that the spin hasn't entered.
Yeah.
We're just out of the spin cycle.
I mean, we have great people there commenting and being critical of things, so we don't have to complain. Recently Jack West on your podcast.
Yeah, Jack West and
many others, but you're right. They spin. Yeah.
So that's important, and then I think this drug dosing point is very important. It'll affect everyone in all specialties. I think people will have to look at it in. You included heme malignancies in your paper. You included everything, all FDA drug approvals. So unfortunately, that means people will have to. They'll have to think of something else to look into because we've already looked into it pretty exhaustively. Any last points on this before we go to the next paper?
I think it's that was surprising. It was so common.
Yeah, I'm still surprised that it's so common. Even thinking it might be an issue, I didn't think it was that big an issue. But it goes to show you, which is I say in the book Malignant, I always say the root issue is that we have a system where the person who is painting the painting is also judging the painting contest. And so it's natural for the pharmaceutical company to say, we have a new drug. Do I think sacituzumab govitecan is better than chemotherapy? I do. I made it, for Christ's sakes. We made it. And should we test it head to head? Yeah. But you know what? Let me give myself a little GCSF, because I want to give myself the advantage, It's natural, I think. And the only way to fix this is to have some impartial validation of studies. I think this is not a question of reporting or transparency. They could do a better job of that so that more people will be aware of it. But the root problem is you need some third party to look at your trial protocol and say that's an imbalance and that's not fair.
If it wouldn't be granted approval, the incentive would change immediately.
Yes, right. It would change overnight. So those are two good papers. What's the next paper that we've had?
We have a lot under review.
I know that's forever under review.
What we had recently, tebentafusp, what maybe
I think
we
we talked about that in
covered
one of the prior episodes.
already.
Yeah.
We have some papers. We can't spoil some.
Okay. Yeah. We
them,
don't want
yeah,
to talk about
yeah.
the ones that are under review. Do we have anything accepted that's not yet come out?
Not yet, no.
No. Okay. Well, that's good. All right. Well, we have a lot of interesting things, and
We have duration of PFS with Alison. Alison, what? Yeah.
yeah.
Duration of PFS.
Oh, yeah, yeah. Have I talked about that on the channel before?
I don't know.
I guess the idea of that paper was to take, you know, you can pick this HCC example. Sorafenib in the SHARP study was sorafenib versus placebo. Then in the Lenva was Lenva versus sorafenib. And then in the atezobev versus sorafenib. But here now we have three arms, sorafenib, 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 and metastatic unresectable HCC frontline. Metastatic unresectable HCC frontline. 3 trials, spanning maybe 15, 20 years. You would guess that the most recent study that had people, you know, with the best imaging and the best knowledge of sorafenib
And supportive care.
and the best supportive care, they would take sorafenib the longest amount of time. And the first study, which had the worst supportive care, they were learning, they never knew sorafenib in HCC, they never know what it was like, they didn't even know if it would work, so they don't really know if they can give it, would have the shortest duration of treatment. But what you find is the opposite. It's 5.7 months in SHARP, it's 3 point some months in Lenva, and it's like 2 point some months in atezobev. Basically, it's like atezobev versus somebody like a whiff of sorafenib in the air, and the sorafenib's gone. So why does it get shorter? And so Alison Haslam, you and I, have a paper in the International Journal of Cancer, where we look at every single time a drug like that has been in the experimental arm and the control arm, and we find not infrequently it has this pattern, which is that the best performance of the drug was in the first trial. In other words, by best I mean the longest duration of treatment. And that's also a bit odd. I gotta say that's a bit odd that would be happening. What do you think about this?
No, again, it's an interest. I think it's also interesting that you have to consider the trials that were done, just not one trial. I have a kind of Bayesian thinking of trials. I don't know,
Yeah.
but look the portfolios of trial. How many trials were conducted? Do the treatment duration change or not? I think that's really a part of critical appraisal. Not just take one trial and study it for hours. You have to know the context of the trial.
Yeah, I think that's something that, you know, as a journal club tip, which is you're presenting a new drug in the second line, bladder cancer, fourth line, whatever. Make a table of all the drugs in that setting. You're presenting response rate of teclistamab. Okay, make a table. Response rate of teclistamab, cilta-cel, and all the other drugs that came before. What was Dara like? What was Velcade like? And also, then correct the denominator.
Melphalan.
And melphalan, sure. And melflufen.
You made this point, yeah.
Yeah, melphalan. Yeah,
And melflufen.
And melflufen, which
We
is
made this point.
totally different drug, obviously. Melphalan and melflufen, which is a novel.
Yeah.
One is an old drug.
We had the paper.
Yeah, we had the paper on that. But you make a table of all this, and then the cilta-cel, you have to correct the denominator because they don't use the intention to treat denominator. Correct it, and then look at it. And then also include in your table, you know, grade 3 pneumonia, pneumonitis. You know, include infection in your table. You'll learn something, okay? So I think critical appraisal is never a study in isolation. It's also in the context of the treatments for that disease. And I think it's also in the context of the broader portfolio of clinical trials across diseases. So the lung cancer people are not wrong to learn from breast cancer, but they're just wrong in what they're learning because they're learning the wrong things. The PATSR didn't work over there, and it ain't gonna work over here, and you gotta validate it here too. So anyway, but I think that's important. The paper with the dean, have we talked about that on the show?
Oh yeah, we did.
Let's talk about
In a way, you talk about before when you did the video when you calculated the.
was and the you know so
maybe it was a osimertinib.
I thought the first time, I think it was either that or that Dean Bajoran paper for like Pembro and bladder cancer.
Oh yeah, that's right.
Yeah. You explained the idea.
So the paper was basically to look at all the adjuvant trials, adjuvant registration trials from 2018 to 2022. So you have maybe 12 approval. You have Adora. You have olaparib in breast.
Yeah.
You have basically
and checkpoint inhibitors and a number of things.
Yeah.
So these are all new drugs in an adjuvant setting.
Yeah.
Okay.
And so at that time, so we studied all the characteristics of the trials. So at that time, they were only based on improvement on surrogate endpoints. soon after, the overall survival benefit from olaparib in breast came out, but at that time, on the 12th, it was just improved
DFS or
GFS,
EFS.
EFS, etc. And what we calculated was the cost to avert one event for each of the drug.
Which is a simple calculation.
Yeah, maybe explain it.
Yeah. So basically, all of these drugs are added to adjuvant treatments. So we've surgically removed the tumor. We're trying to increase the curative fraction. They all have a primary endpoint of some composite time until relapse or new disease or death, whichever comes first, which probably is new disease or death for most people, for most of these settings. And all we're saying is how many people do you have to treat and how much money do you have to spend to prevent one primary endpoint event.
Yeah.
And that event is either a DFS event, an EFS event. And it's important calculation because adjuvant treatment always overtreats someone.
Yeah.
It also always undertreats someone. It's an interesting type of treatment because without the adjuvant treatment, some people are cured and some people will have the disease recur and they will die anyway. And with the adjuvant treatment, all the people who are cured already are going to be exposed to a drug they didn't need because they were already cured. And some people are still going to recur and die, and they were also exposed to a drug they didn't need, because unless it extended their survival, we don't know. But we also exposed to a drug that didn't cure them, and only a tiny fraction of people are getting the benefit. So you're overtreating a lot of people to flip a few coins, to flip somebody who was not going to cure and to be cured. And so it's important. So you get Amazon here too. You get packages here in Switzerland.
Yeah, it's not Amazon.
It's not Amazon, huh? Even their tentacles haven't come here.
Switzerland is maybe the only place in the world where Amazon is not making.
Really?
Yeah.
I wonder why that is. Well, Amazon is a predatory company. Obviously, they sell you things at low prices, but in San Francisco,
I'm not specialized in.
I'll tell you something. I gotta say this: in San Francisco, when you are driving, one lane of every road is the Amazon delivery vehicle. They come and they park, they double park their car.
Yeah, I saw that. Yeah.
They need to start paying some tax for this because they keep parking their car, and you can barely pass some of these streets.
I saw that. Very often.
It's ridiculous.
Very often, yeah.
Everyone lives by Amazon.
Yeah.
God forbid people go to the store and purchase things in person. You could get sick from that, apparently. I don't know why they don't do it. Okay, anyway, back to what we're saying. So we have just covered the drug dosing paper, the GCSF paper.
So we were just talking about the cost per...
The cost per... We're already past that. We're on the cost per thing. The Edine paper. Okay. Oh, you're getting to the punchline.
Yeah, the punchline is that the median cost to avert one event was $1,600,000.
$1,600,000, $1.6 million bucks.
Yeah.
And the reason that's an important number is it's pretty high.
It's pretty high.
It's pretty high. And,
is it sustainable?
Yeah, I just don't think it's sustainable. Somebody's going to come along and say, that each of these DFS events averted is adding 20 years of life to people in the future. But the counter argument to that would be is, where'd you get that number from? You just made that up. Because, of course, many trials have not even reported OS.
Yeah.
You don't even know that to be true. So you're assuming something that's not in question. And cost-effectiveness analyses will assume something that has not been demonstrated. And they will yield probably a different result. But this is why this is to some degree, it's a purer result than a cost-effectiveness. And here's why. Everything that goes into our calculation is a measured value. It really costs that much money, and it really averts that many events, you know?
Yeah.
Whereas when you do a formal cost-effectiveness analysis, you're going to have to impute what you think the life years added will be, and you'll just be basically making up a number because you don't know that to be true because you've never measured it.
I think that was an important paper and it was positively welcomed. Yeah.
It was well received, people...
It was covered in ESMO, I think, ESMO press release.
Oh, good.
Yeah, and the drug dosing was covered also.
In Medscape?
Yeah.
Good for them.
Yeah.
And they covered it critically.
They covered it, they asked some question, I answered the question, and...
These days, a lot of these papers are getting covered. It's always surprising me because I don't even know what's coming, and I just look and I see, it says, we spoke to the author, Alison Haslam, and we spoke to Timothee, or sometimes it says we didn't speak to anyone. We just take excerpts from the article.
Yeah, that's right.
we didn't reach out to the authors. We don't bother them. We just copied and pasted parts of their abstract. But that's good.
That's good.
The
VK Prasad Lab.
Did they put that in there?
No, maybe, but
well, it's a
real lab advertisement.
Yeah. Well, are we looking for people? We have a lot of people now, and we have a lot of projects, but. If somebody really wants to do oncology policy, they should reach out to us. We have our Zoom meetings. And in the Zoom meeting, I always see you with this background.
Mm-hmm.
And now here I'm finally on this side of the
Yeah.
camera. I'm
And
finally
soon we'll be in
on the other side. Soon we'll be back in
California
California.
again. Yeah.
So, what any other thoughts in this space? I mean, I don't know, to summarize maybe some of the broader themes that we've been talking about. We'll come back to the Malignant book series. But I think, the work that we've been doing for the last, maybe that we can summarize some of the work we've been doing broadly. Well, this all got started by a podcast plenary session. You reached out to me, and you had read CheckMate 067, the Larkin melanoma study.
Yeah.
And you were concerned with the rates of censoring because nivo-ipi is very toxic.
Yeah.
And now I think we've been on that issue a lot, and we've proven that it has a high rate of censoring. And you had a paper in the European Journal of Cancer as well, where you imputed different values. And basically you find that, with some very basic assumptions, that the whole thing will tip.
Absolutely. It was in GCE, Journal of Cancer Epidemiology.
Yeah.
Oh, yeah. Same letters, but and yes, there were high rates of censoring of the quality of life questionnaires in one arm as compared with the other, in the combination arm of the CheckMate 067 trial. And we imputed this missing data, and we found that there could be a detrimental effect in the combination arm.
Yeah, and it's just hidden by the fact that there's a lot of censoring. And then we've done this work, and then we have some unpublished work that I presented recently at a UCSF conference. One of which is very provocative, and I think hopefully it'll be well published, and so that people will be able to consider that argument. And some of the other things are, I think, also provocative. What's the uniting theme across all this work? I mean, people call it meta research. What are we doing here? What is this work we're doing?
For me, it's really the core thing is patient advocacy. I would say. What I mean by patient advocacy is what is the best for patient? How can patient be better off? How can patient be protected from toxicities or have less toxicities? And there is also something related to fairness or truth, or you call whatever. But we have to be fair with people. We can't cheat them. We can't sell them something saying it's wonderful, and maybe it's wonderful for some, but we have to be fair, I think. And so for me, that's really the core motivation about clinical practice, and to be really fair in our way of practicing medicine and talking with patients and the families. And
I think that's interesting because to me, clinical epidemiology and evidence-based medicine, they get all sorts of attacks and all sorts of misconceptions about them, but they're really a technique to ensure fairness. Fairness, one type of fairness is that, we actually rely on data and not someone's opinion. That's a certain type of fairness, because in the old days, it was just the opinion of the oldest person with the most gray hair, or the person who's the most forceful at the meeting. That's obviously not the path to truth. So evidence-based medicine is seeking, I think, truth and fairness. And then in these cases, the for-profit incentive has swept oncology. And if you look at a lot of checklists, you'll say, oh, it's randomized, that's good. It's a trial, that's good. It has a lot of people in it, that's good. Good. But there are all these other ways that bias has inserted itself, and you need to use clinical epidemiology, evidence-based medicine, and meta-research to kind of point those out to other people so that there can be awareness of that. And to try to think about how we can minimize that, because what we care about is truth and fairness. On the issue of fairness, the other thing I think that's unfair is that health is both a very important product for people. In fact, I would even say sort of a human right to some degree, like healthcare that actually makes you better off, I think is a human right. But it's also a financial product. It makes a lot of people rich. Yeah. And the people who think of it as a financial product, it's not as important to them that it makes people better off as long as you think it does, and we can sell it. And that creates another bias in the space, which is that if you think it improves healthcare and we can sell it, we'll sell it. And I think that's why some egregious examples get all the news, like the aducanumab, Alzheimer's drug, because everyone can see, you never proven to me it works. Yeah. The harder places are, oncology, which is a niche space. We have to know a lot to even kind of probe it. And then I think the other ones are, some of these broader public policy issues where people have a lot of emotion, particularly with COVID-19. Where they just don't always think very objectively about it. That's a tough place to go. The aducanumab, it's interesting to me. I agree with everyone who's been critical of aducanumab, but we don't need as many of you there. We need a few to spread out, cover the other topics. There's some other problems out there.
Yeah, I think absolutely right.
And then I guess the last question is, you've been in the U.S. and Geneva and Europe, and now your year is almost coming to an end. What would you say similarities, differences? You followed me in clinic.
Yeah.
Maybe you could talk about clinic too.
So clinic, I would say a lot of similarities. A lot of similarities in the way you interact with other specialties, in the way you interact with patient. There are some differences, but I didn't felt it was so different. And what I learned a lot is all the research part, and I came mostly for that. And I enjoy it, and I learn a lot. I'm still learning. I think you learn your whole life, I suppose.
That's the fun part. Here's what I find different. One, you all have a different uniform. It's very white. It's very crisp.
I didn't get into those details.
Oh, those details. Well, there has to be.
Okay.
You have a different.
The location is not the same.
Location's not the same.
The weather is not the same.
The weather's not the same.
Yeah.
The uniform. I gotta say, the uniform. You're dressed like pastry chefs in a fine New York City restaurant. I mean, you're wearing a starched white jacket. The sleeves are usually cut up because you know what? You're taking infection precautions seriously. Because in the U.S., people are reusing a Patagonia fleece. As far as I don't know if it has MRSA fighting properties, but they're reusing a Patagonia fleece. They're all wearing a laundered outfit, and the institution launders it for you, which I think is not always the case in the States. The hospital had a similar layout. The trainees seemed. I think I know this because in the lecture I heard the pager go off a few times, but I don't think it was as many times as I hear in the U.S. The pager going off. And what do I think? I think that the U.S. has kind of become the hospital has become so frenzied and crazy. You can never focus on your thoughts for a split second, particularly the most junior people who are constantly being interrupted.
Yeah, I saw that. I agree, but honestly, you can have these kind of moments also
in Europe. Yeah. But we've been on rounds where we can't even talk to the fellow because the fellow's getting page after page after page after page, and we're like, what is going on? It's like, give this person like 5 minutes so he can at least get some work done. I don't know. I mean, I don't know what the solution to it is. I think there has to be some sort of improvements there. The burnout issue. Do you all talk about burnout in Europe? I didn't feel like that was a common theme.
Oh, that's an issue. That's talk very often. I think.
No, even in Europe.
Yeah, I think it's the same as
I feel like oncology podcasts, 50% are about burnout and working for pharma, which is probably a symptom of the same root discontent. But I don't think about it often, and I don't even feel it at all. I mean, I think that.
But maybe for Switzerland you have a different view because you came with it was novel and you really enjoy and.
Yes, but I that's all true, and I have very much enjoyed my visit with you all and chatting with the fine doctors here. But I don't feel burnout. I don't think in the States, and I think it's because we define our own research agenda and we're pursuing it, and we're sort of genuinely interested in these topics. All right. Any final thoughts, or should we wrap it up?
No, I'm very happy to make this Swiss Alp episode, and I hope listeners will enjoy.
Yes, and they should check out the papers. Yeah. We'll be back in the States. We're going to finish the Malignant Book Club. I think we have 5 more episodes to record.
Yeah, maybe 4 or 5. Yeah.
4 or 5, and we're going to put links in the description to some of the papers people should read. They should follow VK Prasad Twitter website. It just covers the papers. And we'll be back with Dr. Olivier for more.
Yeah.
So, until next time.
Until next time. Bye.
And on that positive note.
On that positive note, very important.